Okay, so that's blocking. One last thing I want to talk about cell preparation is autofluorescence. Cells, and particularly tissue, can have a certain degree of autofluorescence in it that can confuse your specific signal and lower your signal to background. Here's an image taken at 3 different wavelengths and then combined of a kidney section just for its native autofluorescence. And you can see in every color there's a certain degree of autofluorescence there. You don't want this, definitely. And there's a lot of different things that can lead to autofluorescence. It could be overfixation with aldehydes, especially glutaraldehyde. It could be the effects of xylene from your deparaffinization. And it could be endogenous proteins like lipofuscin, which makes those age spots in old people, right? Well, lipofuscin's inside also in tissue, especially brain. Or red blood cells are autofluorescent, or certain plant pigments. So what do you do about these autofluorescent problems? Well, first you can do a sodium borohydride wash. This is where you take sodium borohydride powder that you can buy from many different chemical companies, including the Fisher side of our company. And then you make it very freshly at 1 mg per mil in PBS, and you wash your tissue or cells with it 3 times 10 minutes. And you can see an example here of how I've knocked down some background in the red on these zebrafish paraffin sections. Sometimes people will treat their sections with Sudan black or to quench the background, or photo bleaching it away with sunlight or strong UV light, but sodium borohydride is usually the best way to go. You can also amplify over the autofluorescence. Autofluorescence is usually fairly dim, but if you're trying to detect a low signal, now the signal to background is a big problem. So amplifying with a streptavidin biotin, or in this case, like I'm showing here, amplifying with a terabyte signal amplification kit. Here on the left you can see some tissue that has a pretty bad autofluorescence in the green. But after amplifying with TSA, I've been able to get a nice clean signal with the Bergman glia on these mouse cerebellum sections. You can see an exposure time of only 40 seconds on the right versus 200 milliseconds on the left. So 40 milliseconds versus 200, pretty much brighter on the right-hand side. And then finally, there's ways to use software to remove autofluorescence. For instance, you can leave one channel open. Here, for instance, I've left the red channel open, whereas the green has a few very specific spots on it that were labeled for proliferating cells with an anti-PCNA antibody on rat liver. I then took our imaging software, like our Celeste imaging software, and subtracted the red background from the green signal. And in the far lower right there, you see the result where you see just a few green spots where the specific signal is actually located. There's also special instruments out there from certain microscopy companies for spectral deconvolution, where single color dye samples are used to make a library and then of dye spectra, and then it subtracts that spectrum from your specific image. Remember, overcoming autofluorescence means more sensitivity to give you a better, more publishable image. It's all about signal to background.